Hello and welcome back. We're still in week 3 talking about the macromolecules or the molecules of life. We're up to proteins, which again, I'm guessing you've heard of before. You have some association of thinking about either fish or meat or eggs maybe providing protein, but perhaps you haven't really thought about what are proteins. So proteins, what they're doing is most of the stuff in the cell that gets doing. So carbohydrates, there's some structure for carbohydrates, but mostly short-term energy. Lipids are most significantly long-term energy and some structure, again, of the plasma membrane. But it's proteins that are the doers. So we're going to hit a little bit in the membrane where we're going to talk a little bit more about some specific proteins, but mostly we're going to talk about enzymes in this class, and that's going to be in a few weeks. So I'll hold up on talking really about what they do, but they're going to see that they're really important in allowing chemical reactions to occur by reducing the activation energy. So a bit more on protein structure. So proteins, the things that you're going to be focusing on for now is the structure of the monomer form and the polymer form. So again, monomers, if we were in class, I'd give you a single building block. Those can be stacked together to form the polymer, which is the protein. So a bit more about that monomer. I now again expect you to be able to recognize functional groups. So you can see that what a single amino acid is a central carbon with a hydrogen and one of its covalent bonds, and those other 4 covalent bonds that carbon will always form. One is going to be a carboxyl group, one is going to be an amine group, and then the other right down here you can see is an R group. So from here on in, I expect you to be able to recognize an amino acid. But what that R group is it's different for each amino acid. So we have 20 different amino acids that they all have that same base with that central carbon, the amine group, and the carboxyl group. But then it's this part that is different. That's that R, or that side chain. And what you can see here from just this picture, that some of them are quite small, some of them are quite large. Some of them are going to be hydrophobic, meaning they're scared of water. And it's not really they're scared of water, it's that water doesn't want to have anything to do with them. Water looks at those hydrophobic molecules without any partial charges, and just like a mean girl in high school is like, whatever. And so these guys are going to be not have any partial charge, where these guys are going to have partial charges, or some even have a full charge being positive or negative. And we're going to see in a moment the charge and the size of the amino acid is going to affect how proteins get formed. So here's another picture. As you guys can see by my PowerPoints, I can never decide which graphic to use. So my response is to put all of the graphics in and see which one you personally like better. I'm not saying anything different. It's just showing you the differences in different amino acids. So we have a monomer and amino acid. The monomers come together. Monomer plus monomer is going to form that polymer. Now, we talked about glycosidic linkages for carbohydrates. For amino acids, they're going to form a peptide bond between the carboxyl group and the amine group of 2 different amino acids. So we're going to spend a long time talking about how this is done and the processes of it in transcription and translation. But for now, you just need to understand these structures. You have a single amino acid that gets bound in a dehydration synthesis reaction to form that chain of amino acids, which is going to be making up a polypeptide. Remember I talked about those root words? Another example, poly meaning many. So polypeptide, many peptides. Peptides, proteins. The bond between amino acids, a peptide bond. Yes, there's lots of vocabulary, but if you can clink it together into the right groups, it's a lot less to memorize. So the key thing now that we're going to be focusing on for the rest of this presentation is thinking about the structure and function. Now, proteins above, well, maybe nucleic acids are the same, but the structure and the functions are so directly connected. You cannot talk about a protein doing its function without knowing that protein has a specific 3-dimensional structure. And so these are these lovely graphics right here. Okay, so the structure of a protein, we're going to talk about in 3 different ways. There's 3 different types of structure you can talk about. The first is simply the easiest, which is the primary structure. That is simply just the list in a line of those polypeptides of one amino acid connected via a peptide bond to the next amino acid. So if I were in class, I would hand you all pipe cleaners and you would just have your single straight pipe cleaner. That is considered the primary structure. What you can also see from this primary structure is there's going to be an amino end and a carboxyl end, based again on the fact that every amino acid has that amine and that carboxyl. So you can watch these videos on your own time. In class, what I would tell you to do was now take that strict that was the primary protein structure, and wrap it into slight curls. That is going to be the secondary structure. The secondary structure that forms in proteins forms because of the peptide bond and the hydrogen bonds that are formed. So I've come back to this slide to point out a few things about those hydrogen bonds forming within the secondary structure. It is based on the fact that this amine group is going to be pulling those hydrogens and those electrons from those hydrogens more towards it, because remember, nitrogens are going to have partial charges. Equally, this oxygen going to have a partial charge, a partial negative charge. This is what's going to provide this hydrogen bonding that we see here. So based on those polar covalent bonds within an amino acid, hydrogen bonds form between amino acids, and they form two major structures, either alpha helices or beta pleated sheets. And you'll need to be able to recognize these structures. So you can watch a video on that. Now we have the next level, the tertiary structure. And again, these are numbered 1, 2, 3, 4, primary, secondary, tertiary, quaternary. So the third structure is now you would have taken your wound up pipe cleaner and smushed it in together, where we've got more formation, more folding in on each other. And now the tertiary structure, it's not based on the hydrogen bonding based on the peptide bond, but rather the tertiary structure is completely dependent on which amino acids are in that polypeptide. So depending on if you have a very large amino acid, if you have small amino acids, if you have an amino acid that has a sulfide, if a sulfur, these will form disulfide bridges, which make for really, really strong bonds. We're going to talk about those in one moment, or I'll give you a reference in one moment, but it's this tertiary structure that's formed based on what amino acids are there and what those R groups are. So this is, they have hydrogen bonding between R groups. There can be actually covalent bonding. That's what a disulfide bridge is. There can be ionic bonding if you have a positive and a negative, or negative and a positive amino acid. So the tertiary structure is based on what those side groups are. And then finally, we've got, so this was to show you it depended on what those side groups were. Then we have the quaternary structure. The quaternary structure only happens when one polypeptide joins up with another polypeptide. So when you have one tertiary structure combined with another tertiary structure. So we see that for some proteins, like collagen, that's what makes your cheeks all plump and your ear lobes, where you get your ears pierced. That's all collagen, and it's actually 3 polypeptide chains. Hemoglobin, that we're going to talk about a bunch, or we're going to talk about in the discussions later. That's a great example that we're going to see where you have 4 different subunits coming together. So this is 4 different polypeptides linked together to form one molecule of hemoglobin. So quaternary structure, multiple polypeptides. So again, I have a whole bunch of videos. This single slide overall is your overview. Can you explain the primary, secondary, tertiary, and quaternary structure? Can you describe what is holding these together? In this case, it's the hydrogen bonds based on the peptide bond and those partial charges. The tertiary is based on what amino acid it's made up of. And again, you can see here those alpha helices and these beta pleated sheets. So again, I highly encourage you to watch all the other videos. Here I've got this summary. Can you give this example of how proteins are coming together and how their structure is going to be absolutely key for it to be able to do its function? And we'll see that more when we talk about enzymes. You also need to be able to just recognize these different levels. So if I were to give you this example in an exam, can you recognize that this is, again, the quaternary structure. Quaternary because you can see multiple polypeptide chains coming together. Here, again, you can see in this case, this is actually one single polypeptide chain. It's got two different colors, and this was actually the SARS virus protease before COVID-19, this slide is from, but we can see different proteins, and this is how, when you're looking, actually some data will show you for COVID-19 data what these proteins look like, and now you can understand what those pictures mean. So again, you'll need to be able to recognize. The last things we talk about, the structure is absolutely important for protein function. So if you make a protein denature, that's the term for a protein falling apart. So you have your active protein and its structure, you can make it all fall apart. All you have to do is break those hydrogen bonds that are keeping that secondary structure together. So what's going to break hydrogen bonds? Well, if your molecules are moving really, really fast and hitting, you can't see me, are hitting each other really closely, temperature will actually denature a protein. And you know this actually without thinking about it from when you're cooking eggs. That's what you're doing. You're coagulating and you're temperature changing. You're inactivating those proteins. pH and salt. pH, you're changing those hydrogen ion concentration. That's absolutely going to mess up hydrogen bonding. Same thing with salts. Salts are ionic compounds, and so that positive and negative will also can screw up the hydrogen bonding, keeping a protein together. Sometimes that can be, that denatured can be actually renatured and can reform. That's particularly true for many proteins for the salts and the pHs. Temperature can lead to permanent inactivation. It depends on the protein and the temperature. Or thing to think about, or one good example, here's a link that you can have for a reference to a movie reference for thinking about protein structure. So perms or coagulation, what is a perm? What are you doing? You are breaking the disulfide bonds and you are reforming them in a new structure to give your hair curls.